So over the past 2 days, you heard a lot about the human diversities and health disparities and how these two are connected to each other. But when we talk about the cancer diversity, the case is a little bit different, because cancers not only carry the diversity from the host, but they also have their own evolutionary diversity added to on top of that. And that's the reason why cancers are so complex, and that's the reason why it's so hard to treat and manage the cancers. So what do? As a cancer biologist, we try to simplify things. So we try to divide cancer into 2 types. Type is the bad cancer, and other type is very, very bad cancer. So bad cancer we call the cancer which are indolent, which means they are very slow growing, so they can be managed easily with clinically. But the very, very bad cancer types are very fast growing, they don't respond to the therapies, and these are mostly lethal cancers. And what happens, what people have observed actually, that people belonging to certain racial ethnic background, they tend to get cancer more often, and particularly the very, very bad type of cancer, and that's the reason they die because of it more often. And prostate cancer is one of the examples where we see the highest disparity in terms of the incidence and mortality. As you can see, if you compare African Americans with European Americans, so African Americans, men, they tend to die 1.7, they tend to get 1.7 times more of prostate cancer, and they have nearly 2.3 times high mortality rate as compared to the European Americans. And why is it so? So the reason is simple. If we try to think in a very simple way, that maybe the African American man, they are genetically or epigenetically or both, somehow they are predisposed to getting prostate cancer, or more appropriately we should say they are predisposed to getting an aggressive type of prostate cancer that does not respond well to the therapies, and that's the reason why. It's not that simple, that's why I put that picture, because disparity is quite complex. It's not just genetics, actually it's the interaction of different factors, and many actually believe this healthcare disparities, they also play a major role. Because of certain factors, they don't get the health care that they deserve. So because I'm going to talk about the prostate cancer, so I would like to first explain how the prostate cancer clinically progresses. So prostate cancer originates from the prostate epithelium, and which can progress either into low-grade PIN. PIN is prostatic intraepithelial neoplasia, or high-grade PIN. And it is the high-grade PIN which eventually actually can progress to prostate adenocarcinomas. First, you have just the localized prostate adenocarcinomas, and after that, it can progress to the metastatic prostate adenocarcinomas. So in prostate cancer, we have very good screening. So most of the time, we can diagnose prostate cancer very early. So in that case, we simply give the local therapy, which is the surgery or the radiation, but there is still a significant amount of cases where you have the metastatic prostate cancer at the time of diagnosis. And for those prostate cancer, you have to give them androgen depletion therapy, which is the first line of therapy. But what we have observed over the course of time, maybe in few months or in few years, most of the cancer actually, they come back very aggressive. And we call this type of cancer, when they come back after the castration therapy, castration-resistant prostate cancer. We do have some kind of second-line ADT and chemotherapy, but most of the time those cancers are so aggressive, so therapy-resistant, they don't get benefited by those other therapies as well. So the major focus has been to understand what causes this transition from castration-sensitive disease to the castration-resistant disease. Clinically, what we have observed, the androgen signaling, and it is very, very important to the prostate cancer, and that's the reason we give androgen depletion therapy as the first line of therapy. But it is not only important for the castration-sensitive prostate cancer, but it is also important for the castration-resistant prostate cancer. And in fact, it has been observed that most of the castration cancer, they continue to express androgen receptors, and sometimes they actually overexpress, and they have some kind of mutation in AR. Okay, when we talk about the cancer health disparities, particularly racial disparities, androgen signaling is also important in the prostate cancer disparities. So if you look at the African American men, they usually have high serum level or prostatic level, androgen levels. They also overexpress AR more, and they carry some kind of polymorphism within the AR. And although there are some contradictory reports, and again it could be because of many reasons which already I just talked about, but regardless, there are several reports people have published that in most of the cases there is a transcriptional convergence on AR signaling. So it seems to be a unifying mechanism which promotes prostate cancer in European American prostate cancer patient as well as in African American prostate cancer cases. And there could be many mechanisms which we don't know. So here I'm going to talk about one of the mechanisms that we are studying, and it is related to a protein called MYB or c-MYB. So MYB is a member of the MYB gene family, which has two other important members in the family, A MYB and B MYB. And it is a cellular counterpart of avian myeloblastosis viral oncogene. It is localized on chromosome 6, and it is actually encodes for a transcription factor protein, and has the usual domain which are needed for a transcription factor. So in a normal basis, basically it is expressed by the immature hematopoietic cells, and it is involved in the lineage differentiation, but it has been detected to be overexpressed in certain hematological malignancies and some solid malignancies. So we started working on MYB long back, more than 10 years ago, when basically we did a comparative genome-wide expression profiling, and we found MYB was one of the dysregulated genes that was overexpressed in castration-resistant prostate cancer. And then when we surveyed the literature, we found the prior studies that showed that MYB was actually amplified in prostate cancer, and the amplification frequency increased in hormone-refractory prostate cancer. And when we surveyed the OncoMind database, so we found at least 4 publications in which MYB was shown to be overexpressed. So then we carried out the immunohistochemistry analysis to study the MYB protein expression, because it's the protein which is the functional entity. But what we found when we did, MYB was actually overexpressed in prostate cancer, but when we compared between European American prostate cancer cases, African American prostate cancer cases, we found the overall intensity and overall expression level of MYB was significantly in African American man carrying prostate cancer. We also looked at MYB expression in cancer cell lines, because it was needed not only to see whether cancer as compared to these two normal or benign cell lines, but we wanted to choose a model system to carry out the functional studies later on. So what we found, that MYB was actually overexpressed in most of all cancer cell lines, and particularly if you compare C4-2, this is castration-resistant prostate cancer cell line, which was derived from the lineage. See here, MYB was more here. So what we did actually to do functional analysis, we knocked down MYB expression in C4-2 and RC cell line. This is a African American prostate cancer cell lines, and at the same time we overexpressed MYB in LINCAP, which is a castration-sensitive cell line. And we could see there was a nice association of the growth, increased growth with MYB overexpression. So when we overexpressed MYB, growth increased in these two cell lines, it was decreased. And then we did a long-term growth assays, which is clonogenicity assays. So we found the similar observation. We had just here. Then we studied the effect of MYB on the malignant behavior of the cells. So basically we studied the two properties, migration and invasion. And as you can see here, MYB overexpression actually induced the migration and invasiveness, while when we silenced it, it was decreased. Then we did this actin staining, and what we saw, the cell lines that was overexpressing MYB had this filopodia-like structure formation, which is a characteristic of the mesenchymal cells. And then we looked at the EMT markers, and we found MYB expression was associated with mesenchymal markers. Okay, in this experiment, what we did, we took those cells which were either overexpressing MYB or had a downregulated expression of MYB, and we cultured the cells in the androgen-depleted media. The cells that overexpressed MYB actually could sustain their growth very well as compared to the cells where MYB was not there or we knocked it down forcefully. So after we observed this thing, we wanted to see what could be the mechanism. So, and we were looking for a starting point. So we thought AR is a transcription factor and MYB is also a transcription factor, and MYB somehow seems to provide androgen depletion resistance, so maybe there is some kind of interaction going on between those two proteins. So we did this co-IP, so we pulled down MYB in one case, in another case we pulled down AR, and then we did immunoblotting for MYB and AR, and our data suggested that there is an interaction between the MYB and AR proteins. And then we did this co-localization studies. See, both AR and MYB localize into the nucleus, and there is significant co-localization in the cell lines as well in the tissue sections also. And then we wanted to see this MYB and AR interaction, whether it persists when we culture the cell in the androgen-depleted media or when we treat the cells with enzalutamide, which is an AR antagonist. And as you can see, that is the case. MYB and AR continue to interact when we grow the cell lines in androgen-depleted media or when we treat the cells with enzalutamide. So we wanted to look for the functional implication of this interaction between MYB and AR. So again, we looked at the MYB and AR localization when we grow the cells in androgen supplemented media and androgen deprived media. So you can see here, these are the MYB expressing cell lines. So when we grow them in androgen depleted media, actually AR is sustained in the nucleus, which is not the case in LINCAP. It comes out of the nucleus. So here you see mostly the cytoplasmic exchanging, but here MYB is in the nucleus, and here you can see the co-localization between the MYB and AR. And the similar observation we had with the other cell lines. And then we confirmed by examining the expression of AR by fractionating the protein in cytoplasmic fraction and nuclear fraction. Okay. Okay, so after that we studied activity. PSA is a target of androgen receptor. So we wanted to see what happens to PSA promoter activity in MYB-overexpressing cell lines or when we knocked it down. So here you can see the transcriptional activity of MYB is sustained in MYB-overexpressing cells under androgen-depleted condition. After that, we performed this in silico analysis. We wanted for the MYB-binding site in the PSA promoter, and actually did find one MYB-binding site in the PSA promoter, which was very close to the AR elements. And then we did this chip, and we confirmed that MYB actually was binding to the PSA promoter. Not only that, presence of MYB somehow augmented the binding of the AR receptor to the PSA promoter. Then we wanted to see, okay, so MYB overexpression provides androgen depletion resistance. So do we really need AR, or MYB can actually compensate for the AR loss? So we silenced AR, and then we tried to look for the PSA expression as well as the growth of those cells. So actually our data shows, although MYB binds directly to the PSA promoter, but AR is actually needed for the PSA expression as well as the androgen castration-resistant growth of the prostate cancer cells. So as a biologist, we can do everything in vitro, but unless we do it in vivo in mouse model, nobody's going to believe it, and it's very hard to publish a paper. So what we did, we tagged these cell lines with luciferase so that we can image those tumors when they form tumor non-invasively. So we took this LINCAP cell line, which is low MYB expressing and Lincare MYB, which was MYB-overexpressing cell line, and then C42, SCR, and SH-MYB cell lines. And we directly implanted those cells into the mouse prostate. In a subset, we actually castrated the patient, so we wanted to see what happens to the castration after the growth of the prostate tumor once we castrate the mice. And this is what we saw. We saw the tumor that were overexpressing MYB, they grew faster in both the cases, and when we silenced it, actually the growth was reduced. At the same time, when we castrated those mice, you can see the MYB overexpressing mice continue to grow, while MYB non-expressing cells, the tumor kind of became smaller. They did not grow further. And not only that, we also look for the PSA profiling. What we found that PSA relapse also occurs when the tumors become castration resistant. And then we also looked for the survival of the mice. Here you can see the MYB overexpressing prostate tumor had the worst survival, although the castration provided some benefit, but still it was not it was still bad as compared to low MYB expressing cells. So with that, I'm going to summarize my talk. So what I have shown, our finding tells us that MYB is overexpressed in prostate cancer, and its expression is relatively higher in African American prostate cancer. And MYB promotes growth, aggressive behavior, EMT, and androgen depletion resistance in prostate cancer cells. MYB is a novel interacting partner of AR, and it sustains its ligand-independent nuclear localization and promotes its transcriptional activity, androgen-depleted condition. And in mouse model, we showed that MYB support castration resistance and PSA relapse, and it is associated with poor survival in mice. So there are several questions that are still remain to be answered, and this is what we are currently pursuing. So we are trying to find out why MYB is overexpressed in prostate cancer, especially in African American men, whether it's a genetic reason or some epigenetic reasons, or it's both. And then we are trying to study, does MYB and AR crosstalk lead to the change in their genomic occupancy, leading to some kind of transcriptional reprogramming? And then trying to understand what domains of MYB and AR are involved in the crosstalk between the two proteins. And in addition to, we are also interested in knowing what are the AR independent functions of MYB in prostate cancer, and how we can target MYB for getting the therapeutic benefits. We are trying to explore different ways for it. With that, I would like to acknowledge all my lab members and the collaborators. These are the guys who actually worked for this specific project. I will also acknowledge the funding support from NIH and Institute. With that, I will take any question that you may have.